Today's topic is on novel approaches in pest control. First, we will deal with the push and pull strategy. So, just let me know. Yes. So, the push and pull strategy, it is otherwise known as stimulo-deterrent diversion strategy. Here, you will plant, like we know here, the main crop is sorghum, and along with we have planted the desmodium. So the pull component and push component, they have different works, like the volatile chemicals that are produced by the border plants. They will attract stem borer natural enemies from the sorghum plant, we can say. And simultaneously, if we consider the desmodium plant, the volatile chemicals produced by the intercrop desmodium, they will attract the natural enemies, and as well they will repel the stem borers. So this is the main criteria of the push-pull strategy, and it is a novel pest control approach. But although this is older approach, and this approach has nowadays been renewed just to manage the stem borer that is a major pest in African continent. So coming to the next novel approach, that is behavioral control. Under this, we'll discuss about what is the semiochemicals and what are the allelochemicals and pheromones. So semiochemical, this is a Greek term that is from semion, that is a signal. It indicates the signal, and that are chemicals that mediate the interaction between different organisms, and insect, they might use semiochemicals to convey messages between the organisms and to mediate interaction between them. So semiochemicals can be of two types, like your one is interspecific, that is allelochemical, or else it can be intraspecific, that is pheromones. So basically, if we go to individual level, so we'll discuss in detail like what are the pheromones, how it is being exploited for pest control, and what are the allelochemicals, how it is being exploited for pest control purposes. So coming to the next point, that is allelochemicals that are mainly interspecific. That means among two species. So these are the chemicals that are significant to the individual of a species, different from the source species. So is the figure visible, everyone? This picture. In this, we can see that semiochemicals, there are two arrows, like one is allelochemicals, that is interspecific interaction, and your pheromones, that is intraspecific in action. So basically, the volatile organic compounds that are presented in the parasitoids, they will attract the parasitoids. They are the chiromones, and as well as we can see and that there are cinnamones also and allomones also. So they are basically all allelochemicals. While the under pheromones, we can consider sex pheromone, alarm pheromone, aggregation pheromone, and other plant signaling to the neighbor plants. So basically, if you consider like what is allomone, allomone gives advantages to the releaser who is releasing it. Like we can say different type of repellent. So that is a kind of allomone. So chiromone that are giving advantages to the receiver. While cinnamones, they are advantageous for both the sender as well as the receiver. And anemones are also there, that is emanating from the non-living substances, and like it will attract your beneficial insects. So that these are the basic classification of semiochemical. And for your exam perspective, you can find like each definition you can get in exam, like allelochemicals, what is the term and how it is being derived, and like what are the classification, which one is interspecific, which one is intraspecific, like that classification you can divide. So the next one is like here you can see it is the flow chart of semiochemicals. So chemical communication when it occurs between the same species, that is via pheromones, it is known as your intraspecific communication. And when the communication occurs between different species, it is known as your allelochemicals. So as I told that pheromones might be sex pheromone, alarm pheromone, aggregation, trail or post marking pheromone, or allelochemicals that might be allomones, chiromones, cinnamon, or anemone. So basically, you have to remember this classification. And like I told that allomones, they are beneficial to emitter, means plus sign indicates they are beneficial. And your plus sign again, chiromone, it indicates beneficial to the receiver. Simultaneously, cinnamones, it is beneficial to both the emitter as well as the receiver. So this is the basic classification given by Carlson and Butenandt. And first time Carlson and Butenandt only isolated the pheromone. So yes, this is the first point, like what do you mean by push-pull strategy? So here is the main crop, and pest population will be pushed from the main crop and it will be pulled towards the trap crop. And trap crop, they won't harbor the population, so because the pest biology won't be completed on the trap crop. Due to the antioxidant effect and oviposition deterrent effect and alarm pheromone producing capacity, and non-host volatiles are also there. So because of these components, trap crops won't harbor your insect pest. But while your pool component is there, like there will be host plant attractant, aggregation and sex pheromone, as well as visual attractant. So the trap crops will be having the pull component because of these attractant type substances. So this is the basic of push-pull strategy. So it was first time discovered by Pike et al. In stem borer and in Mage in Africa. So if we come to the pheromones, pheromones are volatile in nature and they aid in communicating among insects. So they are basically exocrine in origin. Exocrine means through ducts they will be produced, and endocrine means without ducts, means the glands don't have any ducts. So that is secreted outside the body. So they are called as ectohormones. So in 1959, first time German chemist Carlson and Butenandt, they isolated and identified the first pheromone, that is a sex attractant from the silkworm moth, that is Bombyx mori, and they named it as bombicol. So later on, we'll discuss the chemical structure and the chemical name of the different pheromones that are important for exam perspectives. They coined the term pheromone, and based on the responses elicited, pheromone can be grouped into two types. First one is your primer pheromone, and second one is your releaser pheromone. So basically, the primer pheromones, they trigger off a chain of physiological changes in the recipient without any immediate change in the behavior. So as they don't cause any immediate change in the behavior, they are of lesser importance in the pest management perspective. But like they act through gustatory sensilla, caste determination and reproduction in social insects like your ants, bees, wasps, termites, that are mediated by the primer pheromones. And as I told, that these pheromones are not of much importance in the practical pest management perspectives. Similarly, if you go to the releaser pheromones, these pheromones, they produce immediate change in the behavior of the recipient, like they will produce through olfactory sensilla. So that's why they are divided into sex pheromone, aggregation pheromone, alarm pheromone, as well as your trail pheromone. So again, you can divide this into two parts, like primer and releasers, I told. So coming to the next point, that is your scientific name of different insect pheromones that is produced by major insect, and you have to remember these points, like silkworm, it produces bombicol, and gypsy moth, gyplure or dispyrlure, pink bollworm, similarly gossiplure, and cabbage looper, it will produce luplure, and tobacco cutworm, it will produce your spodolure or litlure. And then gram pod borer, it will produce helilure. Similarly, the honeybee queen, it produces queen substance. It is of much importance. So as I told, that Butenandt and its co-worker in 1959, they isolated 12 milligram of pheromone from the abdomen of 500,000 of female, and they named it as bombicol. So it is one of the most important objective or bit type you can find in the exam. Like the chemical say primary alcohol. And coming to different if you compare different sex, like among the male and female, usually the females will produce the sex pheromone, but in case of there are some exceptions also where the males will produce the sex pheromone, like cotton ball weevil, cabbage looper, and Mediterranean fruit fly. And scientifically they are known as Anthracis grandis, Trichoplus yarni, and Ceratris capitata. Similarly, what are the uses of sex pheromone in pest management? So basically, they are used for sampling and detection, like to monitor. And second one is to attract and kill, that is mass trapping purposes. And third one is to disrupt the mating, that is confusion and decoy method. And at last, like we can say, that in sampling and detection, what we'll do, we'll just observe whether the ETL has been reached or not. And if the ETL has been reached, based on that, we can go for different management methods. Similarly, in case of mass trapping, you can trap the female moth so that the mating will be disrupted. And confusion and decoy method, the pheromone, it will mimic the natural sex pheromone from the females. So what will happen, the male, they will follow a wrong order corridor, and the mating will be disrupted. So these are the basic pest management strategies using the sex pheromones. And nowadays, what has happened, using splat technique, that is specialized pheromone application lure technique. These pheromones, different type of pheromones, has been widely utilized in pest management. So the sex pheromone, that is, they are mainly released by females, as I told, and they trigger behavioral patterns in the other sex, and they facilitate the mating. So they are also most commonly released by the females, but maybe in some cases they are released by males also. Like the examples are, in case of silkworm, so these are the 5 important chemical names you have to remember. Like in case of silkworm, that is trans 10 6 12 hexadecadienol, and in case of gypsy moth, it is 10 acetosis 7 hexadecanol. In case of pink bollworm, it is 10 propyl trans 5 9 tridecadienol acetate. In case of cabbage looper, it is cis 7 dodecanyl acetate. In case of honeybee, it is 9 keto trans 2 decanoic acid. So these are the 5 important names you have to remember. In case of your aggregation pheromone, they induce aggregation or congregation of insect for mainly for protection as well as for reproduction and feeding. And if we come to the alarm pheromone, mainly it will act as an anti-predator device, and it is a warning. It will provide to the conspecifics about the presence or attack of the natural enemy. Then this warning elicits different behavior, like the insect might disperse or it will escape to different places. So basically, the alarm pheromone are of terpene, aldehydes, and formic acids or monoterpene hydrocarbons in nature. So in aphids, they are mainly terpenoids. In hemipteran, they are mainly aldehydes, and in ants, they are mainly formic acids. And if you come to termite soldiers, they are mainly monoterpene hydrocarbons. So this chemical nature of alarm pheromone produced by different insects will vary from species to species and as well as from different order to order. So you have to remember this. Next is your trail pheromone. Trail pheromone, they used to mainly find the mate and to utilize the food sources more effectively. So most often you might have seen that ant will go in a line because of the trail pheromone, because once the first ant will move, it will secrete a trail pheromone so that the following ants will follow the first one. So the nature of the trail pheromone is mainly, it is caproic acid that is secreted by Eutermopsis, that is a termite. Then hexanoic and heptanoic acid in case of ants. As I told that the ant trail, termite trail, they are examples of trail pheromones. So is that clear, everyone, till now? Yes or no?
Yes, ma'am.
Okay.
Yes, ma'am.
So, okay, thank you. So, we have now discussed that, okay, alamone, chiromone, and cinnamones are secreted, and also we have discussed the importance of different pheromones in pest management. So what about the different plant volatiles? Because now when the insect will feed on the plants, any kind of herbivore that may be, yes, I think some background noise is there. Can you mute yourself? Okay. So, like we know that there are some different type of herbivores that might be biting, chewing type, like caterpillars, or might be phloem feeders. When they will feed on the plants, in response to that feeding, plant will secrete some volatile organic compounds. So that is also known as your herbivore associated molecular pattern. Like herbivore, once it will bite the plant or leaf, that is known as HAM or herbivore associated molecular patterns. and it will release the effector. Effector means it will release one molecule just to counteract the plant defense system. So based on the identification of the effector molecule and the induced plant volatiles, or as we can say the volatile organic compounds, there are also been different management strategies has also been developed, like the effector molecules. If they are artificially created and spread over the plants, they will also attract your natural enemies. So this is a artificial manipulation of natural herbivory pattern. So this figure, I hope it is clear now. Next is your role of pheromones in biological control. So yes, so pheromone means like allomone is beneficial to releaser, pheromone is beneficial to receiver, and your cinnamon is beneficial to both. So beneficial to receiver means they are mainly beneficial for the predatory insects, or we can say natural enemies. So from the herbivore, upon herbivory, as I told that there will be defensive chemicals, they will be produced, like different types of insect growth regulators or different types of hormones. We can say gibberellin, then cytokinin, jasmonic acid, then salicylic acid, abscisic acid. These are different compounds and different molecules that are produced upon insect herbivory. So these will again attract your natural enemies. That is, in this case, the predatory ladybird beetle is attracted to the volatile compounds. So that's how the pheromones has been widely used, and they have also future potential, and they need further research in biological control. Next is what is the inducible defense in herbivore-plant interaction. So this is, although this is not a recent concept, but many novel approaches has been redeveloped based on this. Like we can say that plant in host plant resistance, you know that plant become resistant because of either certain morphological features or certain biochemical features. So sometimes what will happen, like upon feeding or upon herbivory, the plant will secrete some chemicals that will directly and indirectly provide defense to the herbivores, and directly or indirectly they will attack your natural enemies. So different examples of this type of chemicals are phytohormones like volatile organic compounds, extrafloral nectars, then food bodies, and also domatias. Domatias means they will provide a resting structure to the ants. So like I told that physical and chemical different defense compounds are like wax, trichome, cell wall strengthening compounds. Similarly, chemicals are like repellent, toxic compounds. They will reduce the nutrient value by disturbing the insect digestion. So basically, certain characters are already present in the plant, means they know don't need any kind of insect elicitor to get activated. So that type of receptors are or defense is known as your constitutive defense. While the other type of defense system, when the upon insect attack they will get activated, otherwise they will be inactive. That type of defense is known as your inducible defense system. So here we can see that upon clipping, herbivory, or any kind of pathogen infection, the volatile organic compounds will be produced, and they will provide direct defenses by production of hormone, pathogen resistance, as well as your volatile organic compound production. So this is the basically intraspecific signaling. Intraspecific means within the same species they will signal. And if you come to interspecific signaling, like one infested plant upon herbivory, it will emit certain substance that will make aware the other plant that doesn't belong to the same species. So basically we can say that plants also communicate among themselves through the pheromones and through the defensive compounds. So this is the plant communication system, although it is earlier being known, but nowadays it has been widely exploited for the inducible defense purposes. So yes, the next we can discuss about the antioxidants. Antioxidants means they are chemicals which inhibit feeding in insects when applied on the food without impairing their appetite as well as gustatory receptors or driving them away from the food source. So basically the antioxidants, they are different from the repellents because they will in case of repellent, the feeding activity won't happen, but in case of antioxidant, after feeding they will repel. So this is also called as feeding deterrent, resistant, gustatory repellent, or phagodeterrents. Just be cautious of the synonyms because sometimes you might be confused with the repellents, but it is not similar to the repellents. So if we come to the classification of Antifeedants, they might be triazines, organotin, carbamates, or botanical extract. So triazines means they include mainly your acetanilide compounds, like they inhibit your chewing insect, like your caterpillars, beetles, and cockroaches. Similarly, organotins, they are organic compounds of tin. As their name indicate, they are organic compounds of tin or triphenyltin acetate, that is the oldest known organotin, and it is effective against foliage feeding insects such as your Colorado potato beetles as well as grasshoppers. Similarly, different carbamates, like they are considered as primarily insecticides, but at sublethal doses, they will act as antifeedants. So they will be effective against your Colorado potato beetles. That is, example is your Begon. So here, from each group, each example is important. So botanical extracts, like the extract of Chrysanthemum cineridifolium, it is effective against Glossina, that is Sesefly. Then azadirachtin, it is effective against desert locust. Similarly, the apple extract of the genus Malus, it is producing phenolic compound that is known as your florizin. It stimulates the feeding of apple feeding aphid, but it has antiphedency against the other aphid like Mysis persicae. So it is also known as your apple factor. These are different examples of antiphytants. Coming to the different other antiphytants like azadirachtin against desert locust and other insects, Begon against cotton boll weevil, Preston against cutworm and potato tuber moth. Then similarly, chlorinated diphenyl methyl and triphenyl sulfonium salt against phytophagous insects, organotin against grasshopper, agrotis and Colorado potato beetle, florizin against Mysis persicae, then pyrethrum, that is against your Glossina or Sesefly, salanine against potato leaf hopper, as well as thiocarbamate and phenyl carbamate against beetles, then triazine against cockroaches, copper stearate and copper resinate mercury chloride against your tent caterpillar, then cucurbitacin against leaf beetle and spider mite. So these are different examples of antifeedants against their target species. Next is your insect attractant. So attractants are chemical substances. Basically, they will elicit the oriented movement by the insect towards the food source. Like your antipheedant, after feeding, they will prevent the further feeding by the either the insect itself or its progeny. So similarly, the attractants, they will attract the towards the food source. Like mosquitoes, they are attracted by 500-watt infrared lamp. Insects that respond most to visible and UV region, that is in the region of 253 to 700 nanometer. So like we can say that infrared lamp and UV light trap, they can attract the insect. Furthermore, like your violet blue spectral region, it is most attractive for European corn borer, Australia nobilis. Then there are some oviposition lures and food lures. Like oviposition lure means they will attract the insect specifically for the oviposition, like the female will be attracted maybe. And food lure means both the female and as well as the male will be attracted to the source of the attractant to the source for the feeding purposes. So, for example, we can take p-methyl estophenol. It is attractant to the rice stem borer. Hexalure, it is attractant to the pink bollworm. Metaldehyde, it is attractant to the snail and slug. Cinegreen and cinalbin, it is attractant to the Plutilla species. So these are some of the important examples of attractant. And if you come to the food lures, like Aedes species, the food lure is lactic acid and carbon dioxide. Bark beetle, the food lure is terpenes. Then carrot fly, the food lure is phenylpropanoid, as well as the Chafer amphimalon, the food lure is amblure. Similarly, for house flies, sugar and molasses can be used as food lures. And in case of Japanese beetle, the geraniol is a compound that can be used as a food lure. And cinegreen, cinnamaldehyde, as well as methyl eugenol, they can be used as food lure for cabbage butterflies, spotted cucumber beetle, and oriental fruit flies. So these are some of the important examples. Next is your different types of repellents. So repellents, they are substances that induce avoiding movements in insect away from their source, or the chemicals which prevent the insect damage. Means before landing on the food surface, they will prevent they will just repel the insect. So they are mainly following types, like your physical repellent, auditory repellent, barrier repellent, visual repellent, excitatory repellent, feeding repellent. So physical repellent means they are also known as contact stimuli repellent, because after getting in contact with the plant only, they will repel. For example, dust, granule, oil, spine, wax, and they mainly influence the surface texture of the plant by producing a disagreeable effect, and that will just deter the tactile sensor of the insects. So they will deter, and they will be prevented from landing on the host plant. Next is your auditory repellent. Means they will employ sound to ward off the insect, like the recorded sound of supersonic vibration of bat that is used to repel the cotton bollworms. Then next is your barrier repellent, that is painting of oil tar as bands on trees and putting messes will act as a barrier repellent. So, like insects are normally attracted to white lights and but yellow color lights that has a repellency effect. So these yellow color lights, or else we can say yellow sticky traps, that can be used as visual repellents. Next is your excitatory repellents. The chemicals such as pyrethrum, that is BSC, DDT, that would excite mainly the insect site by stimulating the sensory nerves and force them to leave the treated surface. Next is your feeding repellent. So the chemical substances that inhibit feeding on the insects. Oil of citronella extracted from the lemongrass, that is Andropogon madras. It contains citronella, citronellol, then geraniol, baranol, and terpenes. And these compounds have repellency against your mosquitoes. So these are the basic classification of different type of repellent with their suitable examples. Apart from this natural repellents, there are certain repellents that are mainly like we can say that are mainly synthetic in origin. So list of synthetic repellents are like benzyl benzoate, Bordeaux mixture, creosote or 4,6-dinitrocreosol, then diacetyl phthalate, dimethyl phthalate, then N-diethyl M-toluamide, or we can say DEET in short. So benzyl benzoate is effective against mites, as well as Bordeaux mixture is effective against foliage feeders, creosote is effective against chinch bugs, and diacetyl phthalate is effective against cattle fleas. Dimethyl phthalate is effective against mosquitoes, and similarly, DEET also is effective against different type of mosquitoes as well as flies and fleas. Next is like we know that insects, they inhabit different type of endosymbiont in their gut. So that microbiome based insect pest management can also be utilized as a novel source of IPM. So in this, under this, we can consider the symbiont elimination or replacement. Like you have to, if you will replace the gut endosymbiont or you will eliminate it, so the insect will be controlled or dead. Then next is the genetic modification of symbionts, mainly through the paratransgenesis process. Next is your incompatible insect technique. Like if you will, like we can say one mosquito species that is Wolbachia positive, and if it will mate with Wolbachia negative, so it will lead to non-viable offsprings. So Wolbachia is mainly can be used for the incompatible insect technique purposes. Next is your beneficial microbes for mass-reared insect for autocidal program. Like the beneficial microbes, it can be incorporated into the insect population and they can be ultimately used in the autocidal programs. Yesterday we have discussed what is the autocidal program, that is the sterile insect technique, and we can say it has firstly been used in Cochleomia hominivora. Next is your different microbial semiochemicals, because like I told that insect they will, and plants as well as natural enemies, they will produce different type of semiochemicals. Apart from these, the gut microbes also, they will produce some microbial semiochemicals. So that can also be used in pest management prospect. Similarly, the microbial insecticides, like we discussed already, the NPVs, other viruses and protozoans, and entomopathogenic fungi, as well as the bacteria like your BT, that can also be used and as a source of plant protection. And the plant incorporated protectants like your transgenic plants, they can be used in this approach. So next is like what are the potential application of insect symbionts? Insect symbionts, as I told, they might be found in specialized compartments, like we know that bacteriums. Like if the symbionts will lie freely inside the insect body without any organ, so we can say that one is they will harbor or they will stay inside the bacterium, and one is they will stay freely in the hemolymph. So either they will stay in the gut of the insect or in the tissues outside the gut, that is your fat body, or on the cuticle they will stay. So targeting these symbiont interaction can have broad application in pest control or pest population, because they will provide your pest control as well as pathogen control, and also they will provide immunity to different beneficial insects. Like we know that honeybees also, they inside the honeybee gut also different type of microbes are there. So if the microbes can be manipulated, the health of honeybee can be improved. Next is your in vitro growth of different symbionts also can be done, and ultimately the insect microbes can be targeted for biotechnological application. Next is like your paratransgenesis based approach. So paratransgenesis means the symbiont they will present inside the gut of one insect. They will be engineered for the expression of an antipathogen effector molecule. So what will happen after the delivery of the engineered symbiont, like genetically engineered symbiont into the insect, and the antipathogen molecule they will go and colonize in the midgut and reproductive organ of the insect, and it will lead to the production of antimicrobial peptides, and ultimately the immunomodulant molecules will be suppressed. So what will happen, like suppose a mosquito, it is and plasmodium association, and plasmodium is already being developed inside the anopheles mosquito. So due to the antipathogen molecule, the plasmodium development can be deterred. So this is a paratransgenesis based approach. It is a novel approach that can be used for vector control, we can say, like disease vectors, and as well as your different plant diseases or vertebrate disease control we can use. Similarly, Wolbachia-induced method, like I told, that the trans-infection of Aedes aegypti embryos with Wolbachia cells, like Wolbachia-infected mosquitoes, they can only mate with Wolbachia-infected mosquitoes with same strain. So the Wolbachia boosts the immune system of the mosquito and competes for the metabolites. So the insects have different type of immune pathways like toll, IMD for the stimulation of defense. So the mosquito hemocytes, it will produce like different antimicrobial peptides like your defensin, sarcopene, and Zika and dengue viruses that will provide protection. So this modulation of viral replication through Wolbachia method can also be utilized for the pest management, or we can say vector management purposes. Next is your we have already discussed some behavioral chemicals, then we have discussed endosymbiont related novel pest management approaches. Next is your like how to understand the insect physiological systems and correlating it with insecticide mode of actions. So we know that insects have different types of physiological systems like nervous system, digestive system, and circulatory system, respiratory system. So the insecticides, different insecticides, if you mark, they have been developed based on the site of action. So like we can say different octopamine receptors and different acetyl coenzyme carboxylase inhibitors, they will inhibit your lipid metabolism. Similarly, the rhinodine receptor modulators, they will prevent energy metabolism, and ecdysone juvenile hormone chitin synthesis inhibitors, they will prevent the growth and development, as well as the sodium channel modulators, chloride channel antagonists or activators, and SHR agonists or activators, they will prevent the nerve action. So you can see how the insect physiology has been correlated with the mode of action and the resistance mechanism. So what can be done here? Like mode of action approach has been followed. There is a window approach. Like suppose you might have heard that continuously you can't go for use of organophosphorus insecticides. In between, you have to alter it with another insecticide. What is the purpose? Because otherwise it will exert selective pressure on the insect nervous system, so that gradually it will develop resistance to that particular insecticide. So if we rotate the insecticide like one mode of action with another mode of action, what will happen? Like the resistance development can be delayed or suppressed. So basically different types of resistance mechanisms like your metabolic resistance, target site resistance, penetration resistance, and as well as your behavioral resistance. All this type of resistance can be suppressed or can be delayed. This is a type of, we can say, a resistance deterring or mechanisms. So through proper understanding of this interlinking systems, that how insect physiology or insect body structure has been correlated with different different targets of insecticide. So that's how this is a novel approach. Next is what are the other progress and prospects of arthropod chitin synthesis pathways, because we know that chitin is being synthesized and chitin is a major part of the insect cuticle. So if in any way, any mean, the chitin synthesis will be delayed or it will be deterred, what will happen? The insect will not be able to develop properly. So one is your different chemicals targeting chitin biosynthesis, that is the diclobenzoate or etoxazole. So these are the chemicals. Then next is your chemicals targeting chitin degradation, that is your allosamidin. Then RNAi or RNA interference means RNA is the, we know that the central dogma, like from the DNA, the RNA will be synthesized, and from RNA, the protein will be synthesized. So if the RNA will be inhibited, so ultimately what will happen? The chitin synthesis, chitin is a protein, so chitin synthesis pathway is deterred. So chitin being a polysaccharide is being mainly synthesized by the enzyme chitin synthase and mainly degraded by the enzyme chitin deacetylase. So these two enzymes are disrupted through the RNAi approach. Next is your transgenic plants. Like we told already that different plants they having different types of enzymes that are having insecticidal properties can be incorporated to create transgenic plants. Similarly, entomopathogens expressing chitinase enzymes also can be used in the pest management purposes. Then like different chemicals that are targeting chitin regulation, like your tebufenozide, halofenozide, and protein disrupting chitin structures like lectins, proteases, and chitinases are also used in the pest management perspective. So whatever the green color ones here in this picture, they are basically the biotechnological approaches. The purple colored ones indicate the biochemical as well as molecular approaches. And these three, the light blue color ones, they will indicate the chemical approaches. So these are again different types of novel approaches that can be used in pest management. So after we have discussed the behavioral control, we have discussed the novel approaches in chemical control, we have discussed the novel approaches in genetic control. After this, the novel approaches in insect taxonomy. So taxonomy means we basically, for taxonomy we need identification of proper species and till the species level, like say. So usually a taxonomist, if key by key he or she will do the identification, it is very time taking. That's how the DNA barcoding works here. DNA barcoding just shortens the time span of the insect identification, so that what will happen, like within a short span of time, that will within with from a even from a damaged specimen also, the whole specimen can be identified. So what is the process here? Like after isolating the DNA, the PCR analysis will be carried out by using the chromatogram, and then the DNA barcoding standard will be standardized. Then it will be submitted to BOLD. That is, BOLD stands for Barcode of Life Database. And after that, the data analysis and validation will be carried out, and ultimately it will be documented. So for this identification purposes, different types of markers are used. Mainly the markers can be classified into phenotypic, biochemical, cytological, and molecular markers. So these markers, they have been used for insect identification since a long time. Next is your insect growth regulators. So insect growth regulators are the insect hormones, because we know that insect endocrine glands, they will secrete hormones, and that will govern the insect growth processes or maturation process. So when we use a pesticide, these insect growth regulators, they will stop insect larvae from developing into biting and reproductive adults. So the larval development, like we know that metamorphosis, it consists of egg, larva, pupa, and adult. So the larva will not be able to develop into the adult. Next is your like growth inhibitors or insect growth regulators. They are products or materials. They will inhibit the life cycle of a pest. So we'll discuss that how the metamorphosis is controlled and how it is being regulated. So metamorphosis, as we know that it is mainly controlled by the brain. So brain, it will there are two glands again, that is corpora allata and prothoracic gland. Corpora allata will release the juvenile hormone, and prothoracic gland will release the ecdysteroid. So they together, they will govern the larva, pupa, and adult metamorphosis. So now what will happen when the corpora cardiaca and corpora allata, they will be under control of brain hormone, they will release your different types of hormones, like we can say ecdysone and next one is your juvenile hormone. So basically these two hormones, they will interplay in the metamorphosis. So any deregulation or any alteration in the titer of the hormone will lead to the alteration in the metamorphosis. So we can say here in the figure, like this is a diptera or housefly. So here the egg, then larva, then pupa, then adult. So pyriproxyfen, it is a chemical or IgR that reduces the eggs laid by the adult, and pyriproxyfen also reduces the egg viability. Similarly, diflubenzuron and methoprin, they keep the larva in the larval stage only, so that its life cycle won't be completed. And if you come to pyriproxyfen, it results in the fat larva condition. In cyromazine, it keeps larva from molting, like the larva, there will be immature molting. Then if you come to the pyriproxyfen, in the pupal case, the flies, they will die when they will try to pupate. So these are different insect growth regulators. Just the example of housefly I have given. But again, you can see here, like insect growth regulators, they may be of two types. That is, one is chitin synthesis inhibitors, another one is insect hormone disruptors. So chitin synthesis inhibitors are mainly belonging to benzyl phenylureas or BPUs, we can say. So they are diflubenzuron or dimilin, novaluron or rimone, flufenoxuron, lufenuron, chlorofluazuron, triflumuron, and buprofezin cyromazine. So these are mainly the chitin synthesis inhibitors. And if you come to insect hormone disruptors, they might be juvenile hormone analogues or egg-dizone analogues. So as I told that basically the two hormones are responsible for the molting and metamorphosis, that is the juvenile hormones and egg-dizones. So if the production and the transportation of the hormone will be disrupted, then the metamorphosis will be deregulated. So the JH mimics and analogs like your jubabione, methoprin, hydroprin, quinoprin, they are just the naturally occurring JH. And similarly, if you think of synthetic mimics of JH, that are pyriproxyfen, phenoxycarb, and there are certain anti-JH also like precocin. They will induce precocious metamorphosis. Like before the time of metamorphosis, it will induce previously. So the ecdizone mimics, like the other analogs, they are mainly plant-based, that is phytoecdizones, and there are also certain synthetic ecdizone agonists like your methoxyfenozide, halofenozide, and tebufenozide. So these two insect hormone disruptors, along with chitin synthesis inhibitors, these are important for your exam perspective as well. Next is your classification of IgR. If you think like there are some natural as well as some synthetic IgRs also. The chitin synthesis inhibitors, juvenile hormone analogues, anti-JH, molting hormone analogues, they will be coming under synthetic part. And if you come to some other natural IgRs like your limonoid, ketones, enhancing, then ethanolic extract, chlorodomidin, as well as agoracin. So these are from your fruits, leaves, and roots, as well as ocimum and custard apple leaves. So these are effective against different pests like Heliothis, Distichus species, Tenebrio, Culex, and as well as Mexican beetle. So next is your certain market available products. So like I told, diflubenzuron, lufenuron, novaluron, then triflubenzuron, chlorofluazuron, as well as buprofezin. These are available in different markets against your Lepidoptera, Coleoptera, Diptera, and Hemiptera specific insects. Then the major successful examples are your gypsy moth, housefly and ball weevil, armyworm, codling moth, and vegetable bitterns. And different names are there, like the trade name is most important, like the diflubenzuron, it's marketed as larva kill, dimilin, diflumerin, astonex, etc. And lufenuron, it is being marketed as lunox, match, and novaluron, it is marketed under the trade name Rimon, Beltora, and Remosta. Then triflubenzuron, it is marketed under the trade name Nemolt or Nomolt, and chlorofluazuron, it is marketed under the trade name Atagon or A. Jupiter. So these are some of the important trade names, successful examples, target order, and name of the important IGRs. So if we come to the juvenile hormone analogs, means they will mimic the juvenile hormone, and so that if we externally apply these products, so the insect will go under molting process in the not designated time. So examples are like methoprin, pyriproxyfen, phenoxycarb, diphenol. So these are effective mainly against Stepanoptera, Hymenoptera, and Coleoptera, Lepidoptera, Diptera orders. So different successful examples I have given here. like flies, fleas, then mosquitoes and whitefly, jassids, aphids, bollworms, and citrus butterflies. These are the important examples. As well as the marketed trade name is Altosid, Precor, Apex, Minex, and Dita for pyriproxyfen, Award, Logic, and Complectorus for your phenoxyfarm. Diphenolane is marketed under the trade name of Aware. So these are some of the trade names that you have to remember. Few trade names from this. Then your market available of JH, similar like hydroprene, quinoprene, the dryprene, ipronafred. So these are some of the examples again effective against the major insect order, that is Lepidoptera, Coleoptera, Diptyoptera, Diptera, and Hemiptera. So the trade name is Enstar, Altotric, and Gencor, Gentrol, or Sterilizer. So these are most important. Then some molding hormone agonists, like molding hormone means ecdysone agonist products are tebufenoid, halofenoid, methoxyfenoid, and these are marketed under the trade name of Mimic, Conform, Interpret, Runner, Prodigy, Grubex, Falcon, like trade name. And molecular formula is not that much importance, just the target order, some successful examples, and what are the market available products of this IGRs that you have to remember for this. So next is there are some advantages of IGRs. That's why they are being widely used. So like the IGRs, they are effective in minute quantities and they are so economical. Like in spite of chemicals, like we have to apply in huge quantity. The IGRs, they have to be applied in very small quantity. Next is your target specific, and they are safer to natural enemies. So when we consider the chemical pesticides, they are sometimes being sequestered in the insects, and ultimately they will go in the food chain through the to the natural enemies. So they are toxic. But the IGRs, they are safer to the natural enemies. They are biodegradable, non-persistent, as well as non-polluting. Yes. Then they are non-toxic to humans as well as animals and plants. They are well fitted into the integrated pest management program, and most importantly, lesser chances of resistance, resurgence, and residue problems are there. Next is there are some disadvantages. Even though it is stage specific, like usually we'll prefer insecticide or control methods that will control all stages of pest, but IGRs, they will only control certain stages of pest. And also they are having slow mode of action. They are not easily available, because you have seen that I have shown few trade names, but if you search for the chemical pesticides, you can find n number of trade names, n number of insecticides. So from this, you can see that how lesser they are available. So then next is their high cost, then unstable in the environment. Like yesterday, we have discussed the different microbial insecticide. Microbial insecticides also unstable in the environment. That's why UV protectants are added to that. Similarly, the IGRs also, they are unstable in the environment. So these are some of the important novel pest management strategies from different parts of the entomology. Like we have taken the example of chemical control, we have taken genetic control, behavioral control, and then insect growth regulators, then we have taken insect taxonomy and insect physiology also. So these are some of the from different parts I have taken different examples of novel pest control approach. And this is not a static topic, because day by day if you search different papers, you will get every day new topics on this, like what are the novel approaches. So you have to go on reading different types of papers from this. So yes, that's all. Thank you.